Welcome everyone to episode 5, Remastered. Today I'll be discussing a very important class of biomolecules called carbohydrates. Now you might know carbohydrates by their more common name, sugars. Carbohydrates are sugars that can group together to make polymer chains. The sugar monomers consist of a carbon backbone with a carbonyl group and numerous hydroxyl groups. Now, a carbonyl group is a carbon atom that's double bound to an oxygen atom. A hydroxyl group is an oxygen atom that's bound to a hydrogen atom on one side and the carbon atom on the other. Carbonyls can be on the ends or somewhere within a chain, and where the location is within the chain helps determine what kind of carbohydrate molecule it is. The carbonyls that are on the end of a chain are called aldoses, while those that are within a chain, somewhere along the length of the chain, are called ketoses. Because carbohydrates are just chains of carbon atoms with oxygen sprinkled in, it's chains of carbon with just the carbonyl group and a few hydroxyl groups, because of this, different carbohydrates may have the same chemical formula. If you think about a hydroxyl group, its formula is basically CH2O. The oxygen and carbon are each bonded to a hydrogen atom. Now, with the exception of the carbonyl, whose chemical formula is CHO, the rest of the polymer is basically composed of these repeating CH2O groups. The carbon in this backbone shares a bond with a hydrogen atom, and the oxygen in the hydroxyl group also shares a bond with a hydrogen atom. So this means that carbohydrates with, for example, 6 carbons in the backbone will share the same chemical formula: CHO for the carbonyl group, and CH2O6 for the rest of the molecule. Now, this general convention where different molecules can have the same structural formula, this is problematic, because the chemical formula doesn't tell you everything that you need to know to identify the specific species of hexose carbohydrate that you're looking at. Besides the length of the chain itself, the other features that differentiate carbohydrate molecules from one another are the placement of the carbonyl group within the chain, and the orientation of the reactive hydroxyl groups along the chain. Hydroxyl groups can be attached to any carbon in the chain, and they can be oriented in alpha or beta positions, and they can share the carbon chain with a number of other hydroxyl groups. This wide variety in structure gives each carbohydrate a unique function and reactive capacity. Just like the proteins that I covered in episode 3, and the nucleic acids that I covered in episode 4, carbohydrates are polymers. Polymers are long chains or structures composed of repeating subunits called monomers. Carbohydrate monomers are called monosaccharides, mono meaning one, and saccharide meaning sugar. There's a huge variety of these monomers, and they can be combined in an equally huge number of ways to create this really wide variety of different, complex carbohydrate molecules. 2 monomers bound together will make a disaccharide, di meaning 2. When a small group of 3 to around 10 or 12 monosaccharides binds together, these are called an oligosaccharide, where oligo means few. You can think of an oligopoly, which is a government that's ruled by a few privileged or wealthy people. Larger carbohydrate polymers are called polysaccharides, because poly means many. Polysaccharides can be composed of a single repeating monomer, or they can be composed of a repeating series of different monomers. The monosaccharides are all bound together with glycosidic linkages, which are covalent bonds formed through condensation reactions. Now, this might sound somewhat familiar, because these glycosidic linkages are formed through these covalent bonds with condensation reactions. They're analogous to the peptide bonds between amino acids and proteins, and the phosphodiester linkages between nucleotides in nucleic acids. Similarly, all of these bonds can be broken through hydrolysis reactions. Now, sugars can be organized according to the number of carbons in their polymer chain, which is given a numerical prefix attached to the -ose suffix. So a sugar with 3 carbons is called a triose, 4 carbons and it's a tetrose, 5 carbons makes it a pentose, like the sugar molecules in the backbone of nucleic acids. The sugar in the sugar phosphate backbone of your DNA is a pentose sugar. This naming convention will continue logically. 6 carbons makes a hexose, 7 makes a heptose, and so on and so forth. Recall how in the previous episode, I numbered the atoms in the nucleic acids as a tool for organizing them and for efficiently communicating where various side groups are in these nucleic acid rings. Carbohydrates can be numbered in a very similar way. To visualize the carbohydrate molecule, it's often drawn as a zigzagging line. Each point on this zigzagging line, that is, each point that connects two straight lines or two line segments, each point is a carbon atom. Each line, therefore, represents the bonds between those carbon atoms. So in simple carbohydrates, the carbon atoms naturally arrange themselves in this linear, zigzagging structure. The numbering will start at the end that's closest to the carbonyl group. So let's say that the carbonyl group is an aldose on the left end of the chain. The carbonyl carbon, which is the farthest left atom in the chain, is then labeled the 1' carbon. The rest of the carbon atoms in the chain are just labeled sequentially going from left to right. So it's pretty simple. But let's make it a little more complex. What if the carbonyl group is a ketose, and it's somewhere within the chain? But it's, in this case, let's say it's near the right side, like it's closer to the right end of the chain, but it's still, somewhere within it. Well, in this case, the 1' carbon is simply the carbon atom on the far right end of the chain, as this is the nearest to the ketose. The numbering would then go sequentially from right to left. Now, in nature, it's really uncommon to see linear structures in carbohydrates that are longer than 5 or 6 carbons. When the backbone gets any longer than this, it tends to form ring structures. The ring is formed when the carbonyl carbon and a carbon that has a hydroxyl group bind together. The oxygen atom in the carbonyl group is electronegative, so by induction it pulls the shared electrons closer to itself. Furthermore, the resonance structure of the carbonyl oscillates between two states, with a double bond between neutral atoms and with a single bond between a positively charged carbon atom and a negatively charged oxygen atom. These dynamic qualities make the carbonyl carbon partially positively charged, and thus able to attract the electronegative oxygen atom in the hydroxyl group somewhere farther down the chain. Through a reduction reaction, the carbonyl is turned into a hydroxyl group, while the oxygen atom that's in the original hydroxyl group becomes an ester between its original carbon and the carbonyl carbon. This is how a linear carbohydrate molecule can interact with itself to form a ring structure. It's important to understand that this reaction is spontaneous, and it doesn't involve the loss of any atoms. Many sugars have numerous hydroxyl groups that they could hypothetically bind to, but the spontaneous reaction will be the one that requires the least amount of energy. The reaction that requires the least energy will be the one that breaks as few covalent bonds and moves around as few atoms as possible. Consider for a moment the hexose sugar glucose. Glucose possesses a carbonyl on its 1' carbon, and it has 5 hydroxyl groups. In a water solution, glucose spontaneously forms a ring structure that's connected to its 1' and 5' carbons. As glucose is a hexose sugar, it has 6 carbons, so where does that 6th carbon go? The 6th carbon will jut up and out of the ring, like that terminal carbon on the ribose sugar of a nucleic acid. All of the hydroxyl groups attached on the linear molecule will then become oriented in one of two positions after the molecule takes on its ring structure, and these positions are called alpha and beta positions. The alpha and beta positions are determined by a hydroxyl group's orientation relative to the plane of the ring. So think of the ring as making like a disc shape, and the hydroxyl groups will branch off of it either above the disc or below the disc. The top of this disc, or of this plane that the ring forms, is the side that has the 6th carbon atom jutting out of it. So that 6th carbon atom, the direction that it goes is by default the upper side of the sugar. A hydroxyl group in the beta position will also be positioned so it juts out above the plane of the ring. A hydroxyl group in the alpha position, by contrast, will be oriented below the plane of the ring, so it'll be poking out on the opposite face of this ring from that sixth carbon atom. As the reactive side groups on the carbohydrates are these hydroxyl groups, and they have a lot of them, and these hydroxyl groups are involved in the glycosidic linkages and all these other interactions, the orientation and the geometry of these hydroxyl groups has a huge influence on how the sugar molecule interacts with other molecules. Glucose can take two forms. You have alpha glucose and beta glucose, and this depends on the orientation of the hydroxyl group on the 1' carbon. For all glucose molecules, alpha and beta glucose, the 2' and 4' carbon atoms have hydroxyl groups in the alpha position, and on the 3' carbon, they have a hydroxyl group in the beta position. Now, alpha glucose has its 1' hydroxyl group in the alpha position, while beta glucose has the 1' hydroxyl group in the beta position. When two of these glucose molecules bind together, they form a disaccharide called maltose. The two glucose molecules within a maltose molecule will share an alpha 1,4 glycosidic linkage, which signifies the type of bond between the 1 and 4' carbons. In this type of bond, the oxygen atom connecting the monosaccharides sits below the plane of the rings, whereas in the beta bonds, the oxygen atom is positioned above the plane of the rings. Virtually all monosaccharides bind together to form polymers through alpha 1,4 or beta 1,4 glycosidic linkages. Long chains of glucose monomers bound through alpha 1,4 glycosidic linkages will begin to twist and form helix structures. Remember how earlier I said that glycosidic linkages were analogous to peptide bonds and phosphodiester linkages? Well, these bonds do share some qualities, but not all. Peptide bonds and phosphodiester linkages are binary. They have two ends involved in the linkage, which are always in the same spot on each nucleic or amino acid. In carbohydrates, however, these hydroxyl groups, which are these reactive binding groups, they can be anywhere along the ring, and they can be oriented either above or below it. So this means that sugars have this hugely variable modular quality. They don't form exclusively linear molecules like peptide chains or nucleic acids. Instead, because sugars have this highly variable nature in the geometry of their hydroxyl groups, they form these really irregular branching structures. It cannot be understated how important the structure of the carbohydrate is for its function. Even something as small as the difference between alpha and beta positions on a particular carbon can be the single trait that differentiates a structural carbohydrate from an energy storage carbohydrate. Consider the following kinds of carbohydrates: starch, glycogen, chitin, cellulose, and peptidoglycan. Each of these molecules is hugely abundant on the planet Earth, due to their ubiquity in almost every group of living organisms. Starch is a sugar that's used in plants to store chemical energy. The starch polymer is built exclusively out of alpha-glucose monosaccharides connected with glycosidic linkages. There's two types of polysaccharides that compose any given quantity of starch. You have the branching amylopectin and the unbranching amylose. Amylose is a simple linear polysaccharide that just forms alpha 1,4 glycosidic linkages. Amylopectin, on the other hand, shares the alpha 1,4 linkage structure with amylose, but it also has branching chains. Every 30 monomers or so, a chain of alpha 1,4 linked monomers will bind to another chain of alpha 1,4 linked monomers, and they bind together through the 1' carbon on one strand and a 6' carbon on the other. Glycogen is a sugar that is similarly used to store chemical energy, but it's present in animals instead of plants. You and I, as human beings, store our glycogen in our liver and in our muscles. During periods of physical exertion, these huge and wildly branching glycogen polymers are broken down into glucose subunits, which are then used for energy. The structure of the glycogen is very similar to amylopectin, except the branching is 3 times as dense. Branches form every 10 monomers instead of every 30 monomers as in amylopectin. And this raw density of the sugar molecules makes glycogen a very effective, very space-efficient energy storage molecule. Chitin is a structural polysaccharide that's found in the harder tissues of numerous animals, including the exoskeletons of insects, the beaks of cephalopods, the scales of fish, and the tongues of mollusks. Chitin is composed of monomers called N-acetylglucosamines, or NAGs, or NAGs, connected through beta-1,4 glycosidic linkages. Because of this linkage structure, the beta linkages, every other monomer is flipped upside down. So the polymer forms a straight line, but it doesn't form a helix, and this allows the polymer strands to be laid parallel next to one another. The chitin polymers then form hydrogen bonds between one strand and the next, and this creates a very stiff, sheet-like structure. The NAG possesses an acetylamine group in the 2' carbon beta position, which forms particularly strong hydrogen bonds with other acetylamine groups and other monomers. These literal sheets of chitin are deposited strand by strand, layer by layer, forming the tough exoskeleton of a beetle, or the sugary wall of a fungal cell. Cellulose is also a structural polysaccharide, except that cellulose exists in the cell walls of plants and most algae species, instead of fungus, insects, and other animals. Much like chitin, cellulose monomers are beta-glucose molecules connected with beta-1,4 glycosidic linkages, and thus they form long, straight polymer strands. Where chitin monomers have an acetylamine group in their 2' carbon in the beta position, cellulose monomers only have a hydroxyl group. Again, much like chitin, these strands of cellulose polymers are arranged in parallel with one another, and they form hydrogen bonds to create these sheets, or walls. These sheets of cellulose are very regular, they're very ordered and clean on a molecular scale, because the covalent and the hydrogen bonds that form between them create this very clean, grid-like pattern. Peptidoglycan is a third structural polysaccharide. Bacteria have cell walls, but because cellulose and chitin are difficult, if not impossible, for most bacteria to produce, they instead use peptidoglycan. Peptidoglycan is a complex polymer. Each monomer is composed of 2 different sugar molecules, bound with a beta-1,4 glycosidic linkage. So the first sugar in this disaccharide monomer is N-acetylglucosamine, the same sugar that's used in chitin. But the second sugar in the monomer is N-acetylmuramic acid, which is bound to a chain of 3 to 5 amino acids. So N-acetylmuramic acid is part sugar, part protein. The protein segments from one peptidoglycan strand will form peptide bonds with the protein segments on other peptidoglycan strands, which function like the hydrogen bonds in other polymers to create sturdy, sheet-like structures. Consider the fact that virtually every cell in existence uses either starch or glycogen to store chemical energy. Consider the fact that every plant on the planet is composed of cells wrapped in sheets of cellulose. Consider the fact that every insect is encased in a shell of chitin, as are the cells of all fungi crawling through the soil. Now, while each cell and each insect might be very small, when you begin adding up all of the billions and trillions of cells and the billions and trillions of molecules in the exoskeletons of any given arthropod, and you add up the billions and trillions of individual organisms that exist across the surface of the Earth, the total mass of each group of these carbohydrate molecules is almost unbelievably massive. By weight, these are some of the most common biomolecules in the world. Despite the presence and critical function of carbohydrates in every living cell, every modern cell, it's very unlikely that carbohydrates were actually involved in the genesis of life. Carbohydrates do not contain code-like sequences of repeating structures like a nucleic acid, and so sugars cannot act as a template for their own replication. For that matter, no carbohydrate would be able to initiate its own replication, because structurally they're just too simple to be effective catalysts. The reactive hydroxyl groups can only engage in very simple, limited reactions. Proteins and nucleic acid ribozymes, on the other hand, can come in a variety of very large, complex shapes, and these can catalyze all manner of reactions. And perhaps most fundamentally, the glycosidic linkages that connect carbohydrate monomers can't be formed without a particular suite of catalytic proteins. For these reasons, it is unlikely that carbohydrates were involved in the origin of life. Think about this. The very first cells to emerge were far smaller and simpler than the large, modern cells that compose complex eukaryotes like you and I. Without the complexity of organelles or specialized functions, the energy demands on these first cells would have been relatively minimal. Many theories for the origin of life speculate that these early biochemical systems originated on the inner surfaces of deep-sea hydrothermal vents, or maybe in lagoons close to the surface where they could be warmed by volcanic heat and have access to carbon dioxide and sulfur compounds released by the volcano. And they would have also benefited from the heavy energy input of electrical activity, because a lot of volcanic eruptions kind of stimulates atmospheric electrical activity, like lightning and stuff like that. So all of this stuff provides like this ambient physical energy to these biochemical systems. And if you're talking about very, very, very basic primitive cells lining the inside of a hydrothermal vent, they don't really need to store energy for later, because they're just perpetually saturated with chemical and thermal energy. Because of this, there really wasn't a need at the time to invest in some kind of energy storage. But as we can see, these early cells didn't stay in those hydrothermal vents for very long. They blasted outwards into the depths of the ocean, and they began to evolve. Specialized functions or features began to appear, differentiating the early cells and making some of them slightly larger, more energy-hungry. As the early cells became more and more sophisticated, their need for energy increased. Eventually, the cells would need to find a way to store energy for later use, because their sophisticated metabolic and functional activity consumed more energy than what was immediately available. These cells would also need to utilize some kind of reinforcing framework to support the growing size of their membranes and the biomolecules they synthesized. Carbohydrates are the class of molecule that stepped in to fill both of these gaps, providing both an efficient and effective platform for energy storage, as well as providing structural integrity to the walls of the cell. Sugars are really common as structural components in larger biomolecules. Your body, for example, produces amino acids through a series of chemical reactions that starts with a sugar molecule. It builds off of a sugar molecule. Another example is your DNA. Half of the subunits in the backbone of your nucleic acids in your DNA are sugars. You got ribose sugar in your RNA and deoxyribose sugar in your DNA. But how else are carbohydrates involved in the structure of your biomolecules and the structure of your cells? Well, carbohydrates are very durable, and they're very elastic molecules that show levels of structural organization, kind of, but not exactly like the primary, secondary, tertiary, and quaternary structure of proteins. Recall how a moment ago I described polymer strands lining up beside one another and connecting through numerous hydrogen bonds. Groups of these hydrogen-bonded strands form sheets, and these sheets can either be layered on top of one another, or they can be twisted together to form larger fibers, and those large fibers are themselves piled and crisscrossed to create an even larger elastic sheet. Sometimes the main structure is just a massive, strong fiber. Cellulose, for example, forms fibers that are made out of 70 to 80 polymer strands. These massive cellulose fibers will pile and overlap, covering the cell in a flexible but sturdy sheath. Recall that the structural sugars that I described earlier all share beta-1,4-glycosidic linkages. The structure of the beta linkages makes the fibers very durable, inaccessible to water molecules, and thus insoluble in water, and resistant to being broken down by hydrolysis. This is also why structural carbohydrates are so effective. Cellulose, or dietary fiber, is found in plant foods. Fruits and vegetables are high in cellulose, as are whole grain breads with seeds. The cellulose itself is indigestible, so when you eat foods that contain cellulose, like fruits, vegetables, and whole grains, instead of being digested, it coalesces into a spongy ball or a spongy mass, and this will help hold moisture and moisturize and lubricate your intestines. The wet fiber mass will also assist in smooth digestion and easy, regular bowel movements. This is why they say fiber is so important to a healthy diet. Carbohydrates also play a very important role in cell-to-cell communication, in cell signaling, and cell identity. The cell's membrane is composed of a bilayer of phospholipids, studded with various proteins. I'll go into more detail on lipids and membranes in the next episode, but I need to mention this briefly for now because of the importance it has for glycoproteins. Glycoproteins are a pretty cool sugar-protein combination, wherein the protein is studded within the membrane, and the sugar extends off the top of the protein, reaching away from the surface of the cell. Glycoproteins possess a huge variety of oligosaccharides, which play a really critical role in the proper functioning of your body. These oligosaccharides are used by your immune system to identify cells that belong to the self, that belong to you, cells that are healthy and belong to your body. The cells that don't have these identifying sugars are perceived by your immune system as not self, or not a part of you, and thus they become targets for the T cells, the natural killer cells, and all the other antigen-destroying immune cells. These identifying sugars are not the same for every cell in your body. Different types of cells have different groups of identifying sugars on their glycoproteins. Your neurons, for example, have a different set of glycoproteins than your dermal cells, which are different than those in your muscle cells, which are different than those in your retinal cells, and so on and so forth. For example, when a sperm cell and an egg cell meet together, they'll fuse and become a fertilized egg, which will then grow into a zygote, which will then develop into another individual. But in order for the sperm to fuse to the egg and initiate fertilization, it has to find a glycoprotein with a carbohydrate that it recognizes and can bind to. Namely, it has to find a glycoprotein of its own species. There are many reasons why organisms from two different species can't interbreed, and the inability for sperm to find a recognizable carbohydrate on the egg cell is one of those reasons. If the sperm does recognize the signaling sugar being extruded from the egg cell, then this will lead to a successful binding event. It will initiate the absorption of the sperm into the egg cell, the deposition of the sperm's genetic material, and its combination with the egg's genetic material, and this will then begin the process of the development of a new individual. Besides being used for structural support and cell signaling, carbohydrates are critically important for their role in energy storage. Remember how I said that monomers connected with beta-1,4 glycosidic linkages make for good structural proteins? Well, what about the monomers that are connected with alpha-1,4 glycosidic linkages? These monomers form polymers that are easy to break down with hydrolysis. This is good for a complex molecule that's meant to store energy, because chunks of it can just be readily broken off whenever it's needed, and they can be metabolized to supply energy to the cell. For alpha-glycosidic linkages, like the ones in glycogen, an enzyme called phosphorylase is required to initiate hydrolysis. Your muscle cells and your liver cells have higher than average quantities of phosphorylase, as these are all needed to rapidly break down the glycogen that's stored there. Understand, however, that virtually all cells have some levels of phosphorylase, because all cells need to access their own troves of stored chemical energy. This energy is then used to power all of the cell's various functions, from signaling to secretion of produced biochemical products to basic housekeeping functions like cellular respiration and division. Okay, now you might think I'm kind of dancing around a key issue. How is energy literally stored in carbohydrate molecules? How does the cell access this energy in a usable way? What does it mean to say that there's energy here and it's being stored like a chemical battery? Like, what does that mean? Chemical energy exists in the bonds between atoms. Consider the carbonyl double bond, or the hydroxyl single bond, that exists between an oxygen atom and a carbon atom. The carbon is electronegatively neutral, while the oxygen atom is very electronegative. Because of this quality, the oxygen atom holds the shared electrons in this covalent bond closer to itself, which reduces the potential energy of the bond. This makes the carbon-oxygen bond bad at energy storage. If you watched episode 2, you might remember a metaphor that I made explaining electronegativity with planets and gravity. Imagine some kind of mass, something big and heavy, like a bus without wheels. Now imagine that you're pushing this bus without wheels while it sits flat on the ground. You can get a few friends to help you, but chances are that bus isn't going anywhere. You're not going to be able to move it along the ground. What if you and the bus were, for the sake of hypothetical argument, floating in a dark expanse of microgravity? Your spacesuit has a thruster pack, but it's kind of weak and can only produce the same amount of thrust that you could produce on the surface with your little meat body, like when you were pushing in vain against the grounded bus. So now in microgravity, you're applying the same amount of force to the bus, but in microgravity, the bus will slowly accelerate. If a big purple gas giant suddenly appeared nearby, the bus would be attracted to it, and it would accelerate as it fell into the gas giant's gravity well. In this way, the bus has more potential energy here in microgravity than it does sitting on cinder blocks in someone's backyard. Electrons behave somewhat analogously. I know it's kind of a rough metaphor, but electrons kind of work in the same general way, if you consider the valence shell akin to the surface of the planet. Nearer the surface of the planet, objects will have less potential energy, and they'll feel heavier and harder to move. Similarly, electrons that are held close to a strong source of electronegativity will have a very low potential energy, because it's just hard to get those electrons moving anywhere. So if oxygen has a strong electronegativity, and this is causing the bonds that it makes with carbon to have low potential energy, low chemical energy, then what about the carbon atoms? Carbon atoms are electronegatively neutral, as are hydrogen atoms. In a carbon-carbon bond, like the ones that exist all down the sugar's backbone, the electrons are equally shared between the atoms. Continuing with the gravity metaphor, this is like the potential energy of a large item on the edge of a gravity well. It has a large potential energy here, and depending on which way you push it, it could easily be, just pushed right into the edge to fall into the gravity well, or it could be pushed out of the gravity well. I'm sure there are chemists out there that are just screaming and pulling their hair out, and they're just itching to tear my metaphor apart, but if it helps you understand this concept in general biochemical terms, then that's what counts. Anyways, the alpha-glycosidic linkages of energy storage carbohydrates can be broken to yield metabolically accessible glucose monomers. This is where the phosphorylase and the amylase come into play. These enzymes are part of the protein suite that is necessary to conduct hydrolysis on the glycosidic linkages of glycogen and starch, respectively. The glucose monomer is then consumed in a process called cellular respiration, which I'll explore in a future episode, where the glucose molecules' bonds are broken down. These other molecules include a variety of energy storage carbohydrates, but they consist primarily of a nucleotide called ATP, or adenosine triphosphate. ATP is synthesized from several precursor molecules, including molecular oxygen, an inorganic phosphorus atom, the carbohydrate, which is consumed for energy, and a molecule called ADP, or adenosine diphosphate. The energy that exists in the chemical bonds of the glucose are used to attach the inorganic phosphorus atom to the other phosphorus atoms in the ADP. This produces ATP, which your cells then consume to access the energy in usable ways. ATP will thus release energy when its phosphorus groups have been broken off, and the high bond energy used to power another chemical reaction, or change the conformation, or do really anything else that requires energy. In all of these ways, carbohydrates can provide both structural support to cells and a format for storing energy on a molecular level. As useful as they are, carbohydrates pale in comparison to lipids when it comes to energy storage. I mentioned lipids earlier in this episode, and I've mentioned lipids, here and there in all the episodes before this. And I'll be discussing lipids in much greater detail in the next episode, which will be the last one in this first series on biology and biochemistry. But as for episode 5, that's about it. I hope you learned something neat. I hope I kind of opened your mind to the world of carbohydrates and how cool and dynamic and useful they are. And I hope you're excited for next week's episode. As always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button, share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.